Welcome, and thank you for joining us today for the Investigating the BMI Paradox in Kidney Cancer webinar. My name is Vanessa, and I will be your WebEx technical support host. Before we dive into today's meeting, I wanted to provide some quick information on the webinar platform. All lines have been muted on entry and will remain muted for the duration of the webinar. Please submit questions throughout the presentation using the chat and select all panelists. A moderator will ask the comments and/or questions on your behalf at the end of the presentation. If you require technical assistance, please feel free to contact me, the host, via the chat panel, and I'd be happy to assist you. If you need closed captioning, please refer to the link in the chat panel. We also wanted to make you aware that today's webinar is being recorded. And now I'll turn it over to Dr. Joanne Elena to introduce our speaker.
Thank you, Vanessa. Well, we are in luck for another great talk as part of this Body Composition and Cancer Outcomes webinar series. Today we have Dr. Furberg Barnes, a molecular epidemiologist from Memorial Sloan Kettering Cancer Center, whose research focuses on how modifiable factors influence genitourinary cancer etiology and prognosis. She is, among many things, the co-PI of the Urothelial Cancer Registry, an institutional resource to identify genetic and non-genetic factors associated with bladder cancer risk and outcomes, and is the PI also of an American Cancer Society funded study to determine the impact of post-diagnosis smoking on bladder cancer recurrence. I've gotten the chance to work with her through several grants funded by NCI, including her R01 Body Composition and the Obesity Paradox in clear cell renal carcinoma, which will be providing the foundation of her talk today. Without any further ado, please take it away, Helena.
Thanks. Hi, everybody. So I'm just going to go ahead and jump right in. I have no disclosures to report. As she mentioned, I'm funded by the National Cancer Institute and also ACS. And I'm hoping, I put together this talk to provide kind of an overview of the background and justification for what we're calling the RESOLVE study. This is an NCI-funded R01. It's a molecular epi study that we, the funding started in August of 2019, so we're about a year and a half in. It's a retrospective cohort study focusing on 1,200 clear cell renal cell carcinoma patients. We're interpreting existing CTs for body composition, and we're linking those data with transcriptomic profiling of archived tumor tissue, and ultimately trying to better understand how body size influences clinical outcomes in these patients. And this is truly a multidisciplinary research effort, so I'd like to spend the first few minutes acknowledging my research team. It spans 4 different institutions and several departments. So in Memorial Sloan Kettering, we've got members in the Epidemiology and Biostats department, surgery, bioinformatics, radiology, and the pathology biospecimen core. All the tumor specimens are being sent to Dr. Melissa Troster at UNC Chapel Hill, and for transcriptomic profiling on the nanostring assay. And then our body composition phenotyping is led by Dr. Marina Martsakis at the University of Waterloo, and also her lab member, Mike Paris. Our study is anchored by Dr. Betty Kahn, who we all know as starting this lecture series, and also for her expertise in large-scale studies of breast and colorectal cancer and how body composition influences those outcomes. I'd also like to acknowledge a few other people who aren't directly involved in the R01 who have been really helpful in helping me understand and unpacking the BMI paradox. These include Andy Dannenberg from Weill Cornell, Whitney Robinson from UNC Chapel Hill, Miguel Hernan from Harvard, Mark Perdue from the National Cancer Institute, David Thomas from MIT, and also Jessica Scott from Sloan Kettering. All right, so first I thought I'd provide some background about kidney cancer, because when I first started studying this cancer about 10 years ago, it struck me at how different it is from most common cancers that people study, like lung or breast or colorectal or even prostate cancer. So as I kind of go through these slides, you might be thinking about how it could affect the study of body size and clinical outcomes, and I think that you'll agree that some of these features actually make it easier to study than some of the other tissue types. So kidney cancer is the 8th most common cancer in the United States. Unfortunately, its incidence is increasing, but there are smaller tumors being diagnosed. The most common type of kidney cancer is clear cell renal cell carcinoma, and that's what I'll be focusing on in this talk. It's the most common. It's also the most lethal. It's more common in men than women, and also more common in whites than other racial and ethnic groups. There's been a lot of genomic characterization of ccRCC. It's one of the tissues that was profiled in the Cancer Genome Atlas that I'll get to later. It's characterized by alterations in angiogenic pathways, that's what forms blood vessels, the immune pathway, and also metabolic pathways. So one of the key features that makes clear cell renal cell carcinoma so different and unique is that over 75% of patients who are diagnosed with kidney cancer are diagnosed incidentally. This means that they present with no symptoms, they have no unanticipated weight loss that they can see anyway and report, and they're detected because they go and get imaged for some unrelated reason, like maybe they have some stomach pain or they broke a rib, and then the technician or the physician says, oh, we see a renal mass, but you should go get worked up. So they go get worked up, and then it turns out that they have kidney cancer. So that's one thing that is very different than other malignancies. The other thing that's different is that kidney cancer or clear cell is a chemotherapy and radiation resistant disease. So that means that treatment options are limited. Non-metastatic clear cell renal cell carcinoma is, so these are stages 1 through 3, is treated by nephrectomy, whereas metastatic clear cell is treated by targeted therapies. Oh, whoops, that was some weird formatting going on here on the slide. But metastatic disease is either treated by targeted therapies like sunitinib, that goes after the VEGF pathway or the mTOR pathway. It's also more recently treated with immunotherapy. So it's one of the few cancers that are treated with immunotherapy. The other ones include lung and melanoma, for example. As with other cancers, your stage is directly related to survival. So like I said, most people are presenting with early stage disease, so non-metastatic disease, which is stages 1 through 3. So about 80% of patients present with that. Their survival for 5-year in the localized setting is 81%, but it drops to only 8% once the tumor has metastasized. And somewhere between 10 and 30% of localized disease either relapses or progresses to metastatic disease. There have been pretty, in comparison to other tumors, few risk factors that are strongly associated with renal cell carcinoma. These include cigarette smoking, high BMI, hypertension, and some familial cancer syndromes. Other factors like diabetes, low physical activity, alcohol consumption are suspected, but they're not confirmed. So our group focused on BMI as a risk factor, and we know that from lots of other studies that about 40% of kidney cancer is attributable to obesity, and that's defined as a BMI, just traditional WHO definitions of BMI greater than or equal to 30 kilograms per meter squared. For every 5 kilogram meters squared rise in BMI, the risk of developing RCC increases by about 24% of men and 34% for women. And associations are strongest for the ccRCC histology. Oops. This next slide is a schematic of the putative obesity-related mechanisms through which obesity could increase risk of developing the disease. These include renal hypoxia, lipid peroxidation, altered hormones, hormones that are coming even from fat tissue. And so since these mechanisms can not only induce but also promote cancer, you would think that high BMI would be an adverse prognostic factor if you looked at how it was associated with clinical outcomes. This is not what is seen consistently in literature in clinical-based cohorts. In 2013, there was a meta-analysis of 15 studies conducted by Choi et al. Of over 12,000 patients. And these figures here are the plots showing a bunch of different studies and their pooled hazard ratio for overall mortality and for cancer-specific mortality. So the pooled hazard ratio between comparing obese versus normal weight patients was 0.57. So this shows that there is an inverse association between BMI and mortality, such that patients with high BMI have lower mortality. And same thing was seen with cancer-specific mortality. We followed this up around the same time with a paper that we published in JNCI in 2013. This was a retrospective cohort conducted at MSK. 90% of our cohort were non-metastatic, but what was different, what we did was we focused on clear cell because we know that they have genomic differences than other histologies in kidney cancer. So we wanted to be as specific as possible. All of these patients were treated with nephrectomy. We had BMI at the time of their nephrectomy. And as you can see, 80% of the population was classified as obese or overweight by BMI at the time of their nephrectomy. So these patients are quite large at the time of nephrectomy. So we looked at how BMI at the time of nephrectomy was associated with outcomes, and we found, again, similar to what Choi found, these inverse associations. I'm sorry, somehow these formatting of these slides are not exactly right, but you can see that overweight versus normal weight was 0.73, and obese versus normal weight was 0.59. And what was notable about our analysis is that we tried as best we could with the data that we had to try to figure out, like, address unmeasured, unaddressed confounding. We stratified by certain groups to see if the association persisted by stage, by sex, and by incidental or symptomatic presentation. And no matter what we did, the inverse association persisted. The next thing we did was partner with a group from Harvard who was focused on metastatic patients treated with targeted therapies. So this was a study done by Albiger et al. In 2016, published in JCO. Oh my God, the formatting is really crazy in this one. And what she did was she did a discovery and validation cohort approach. So basically a two-stage study design. So she had over 2,000 people in the discovery cohort, 4,000 people in the validation cohort. And so keep in mind now we're focusing on stage 4 kidney cancer patients treated with targeted therapy. And these are the ones that go after angiogenesis pathways or mTOR pathways. And again, she found the overall mortality hazard ratio comparing overweight and obese to normal weight were inverse. Both in the discovery and the validation cohort. And most notably, this was adjusted for the International Metastatic Disease Consortium score, which is the best sort of prognostication and the most common way to adjust for known prognostic factors. It divides people into low, like poor, intermediate, and high risk. Poor, intermediate, and favorable risk categories based on 6 different factors, including time since diagnosis, some blood factors, and then also Karnofsky performance status. We followed up that work most recently in 2020 by a paper that we published in Lancet Oncology last year. This looked at a bunch of different cohorts. One was the COMPARES randomized trial of targeted therapy. So we basically wanted to follow up what we had found in the Albiger paper. This was a study of 256 metastatic clear cell patients. And here again, we found an inverse association. We also had we put together an observational cohort of 129 metastatic clear cell patients that were treated at our institution with immunotherapy agents, and we found an overall mortality hazard ratio, again, of 0.72. But this became non-significant after we adjusted for the IMDC risk score. There have been a bunch of other studies, a few of them are listed here in the references, that are looking at this in immunotherapy, because since it's a new treatment for kidney cancer patients, the clinicians and patients are really eager to know if and how body mass index, for example, or body size influences clinical outcomes in patients treated with immunotherapy. So with all of that, so now we're seeing that this inverse association is consistent. It's seen regardless of treatment type in kidney cancer. We're seeing it in nephrectomy treated patients. We're seeing it in targeted therapy treated patients, and it's suggestive, I would say, in immunotherapy treated patients. So what does this mean? Is the association causal? Some people interpret it as causal, where they think that excess weight is beneficial in the context of disease. And I would say that this is unknown at this time because we don't know what high BMI represents, and you have not done an analysis to see whether gaining or losing weight after diagnosis influences prognosis. Another option is that the BMI paradox might be driven by unaddressed methodologic issues. So what about disease-related wasting? So remember at the beginning, I told you that most people are diagnosed and they have no clue they had the disease and they do not report any symptoms, including pre-diagnosis weight loss. So there's few people reporting that. The inverse association that we detected in our study persisted even among incidentally detected patients. That's how we try to control for that. And there's no neoadjuvant treatment like there are for some of the other tumors that could induce treatment-related wasting. So we don't have to worry about that happening. Another issue is that collider bias could be at play. And so this is an epimethods kind of term, collider stratification bias, and it basically boils down to sort of unmeasured confounding with selection bias that are present. And so it happens when turns out that you can get unusual associations or paradoxical associations if you condition, if you restrict your analysis to, for example, cases only, and you end up comparing, you only have people who have the disease in your data set. So you're comparing people with high BMI to people who are normal weight, and then you're looking at how it influences outcome. And if you think about that, the people who are obese probably got, at diagnosis, probably got kidney cancer because they're obese. But people who are normal weight and haven't experienced wasting, these people are not frail, like 80% of them are overweight or obese at diagnosis. And even the normal weight patients, we're not seeing a lot of frailty. When I've shadowed Dr. Russo in the clinic, these patients seem, for the most part, very healthy, and they're all healthy enough to undergo surgery. So the normal weight patients, though, got kidney cancer through some other mechanism. And we're not sure what that is, but if they harbor, if they got a tumor that is, or they have some other feature that is more lethal, then it's going to look like obesity is protective, when really it's that there's something that's very harmful going on in a normal weight patient. So it's important to understand and unpack kind of what the interpretations mean and not just interpret the data kind of on the surface. We hypothesized that there might be differences in tumor biology by BMI that could be influencing the BMI paradox. Oops. So what we did was leverage data from the Cancer Genome Atlas. This is an NCI initiative. There are 33 different tumor types that have been like comprehensively genomically characterized. Clear cell, renal cell carcinoma is one of them. MSK was one of the main participating sites and contributed the highest number of specimens. So we used those, 126 of them. We abstracted BMI at the time of specimen collection because that is not routinely, that's not part of the TCGA data set. And then we compared mutations, copy number, methylation status, and gene expression patterns in tumors by BMI. So what did we find? This was, by the way, published in the JNCI paper from 2013. We did not find any differences between BMI and somatic mutations, methylation status, or copy number. But what we did find were significant differences in gene expression by BMI. So to orient you to this slide, you have genes that are differentially expressed over here on the left, and then you've got BMI up here on the top. So normal weight is over here, obese is over here, and genes that are upregulated are red, genes that are downregulated are blue. And so please take a look at the genes, and if you have any favorites that you can help us interpret, we'd be happy to hear about them. The one gene that we noticed in particular that stuck out, that we knew something about, was a gene called FASN, or FASN. So FASN was differentially expressed by BMI. We know that it's a metabolic oncogene. It encodes for a rate-limiting enzyme involved in fatty acid synthesis. Overexpression has been associated with tumor aggressiveness and poor prognosis in kidney cancer. And there's also been studies that show that pharmacological inhibition of FASN can suppress tumor growth in kidney cancer cells. So it looks like if you have an upregulation of FASN, this is a bad thing for tumor growth. And in fact, we found that the pattern in our data was such that FASN was downregulated in the tumors of the obese and upregulated in the tumors of the normal weight. So this led us to preliminarily conclude that tumors of obese may be less aggressive than the tumors of normal weight patients. Next, we tried to validate this, or actually the Albija's group, who we participated with she validated the signal of FASN, BMI, and mortality in 2 different independent cohorts. So she had one cohort where she had 61. Remember, the Albija's paper was the one that was published in JCO, and that was looking at metastatic patients treated with targeted therapy for the VEGF and the mTOR inhibitors. So cohort 1 were 61 metastatic clear cell patients, and then cohort 2 were 165 metastatic CCRCP patients from the IMDC biobank. And what she found was, I'm not sure if you can read everything that's on this slide, but what's shown in yellow is upregulation, and what's shown in blue is downregulation. And you can see that obese patients are more likely to have a downregulation, normal weight patients have an upregulation, and then she looked at how FASN was associated with survival, and so you can see that FASN was upregulated in the normal weight over here, and that FASN upregulation was associated with inferior survival in both of these cohorts. So this is the yellow line here. The next thing that we did was the most recent study that I mentioned, the Lancet Oncology paper, and here we decided to take more of a candidate gene approach. And since we know that treatments for kidney cancer involve the immunogenic and immunologic, those are treatment-related pathways. So we thought we'd see how BMI was associated with each one of those. And so for this study, we had access to several data from several different cohorts. One was the COMPARES trial. These were metastatic patients treated with targeted therapy. We had 93 tumors of metastatic patients from the TCGA. And then we also had 62 perinephric fat samples from non-metastatic clear cell patients. And this was an effort that we did with Andy Dannenberg, where we collected from, at the time of their nephrectomy, not only a piece of their tumor for profiling, but also perinephric fat. And we collected perinephric fat near the tumor and farther away from the tumor. And then we extracted, Andy's lab extracted the RNA from those fat specimens, and then we ran those for gene expression. So what we found was that first we looked at differences in the tumor. And in the COMPARES cohort of metastatic patients, we found differences in angiogenic pathways. And this was validated in the cohort of TCGA patients. So tumors from obese, that they differed by BMI, I should say, and that the tumors from the obese had an upregulation of angiogenic pathways compared to those that are normal weight. And we saw the same pattern in the TCGA. We thought we would see the same thing with immune signatures and immune infiltration, but we did not find that to be the case. We found no differences in tumor infiltration by BMI. But then when we turned our attention to the perinephric fat, that's where we saw some significant differences, that obese patients had an increased infiltration of immune cells in perinephric fat. So this suggests that the tumor microenvironment is important to study, and we might see differences in some kind of crosstalk that's going on between the fat cells and the tumor cells. This is a very hot topic right now. A lot of people are doing excellent work trying to unpack this association across different malignancies. Wang had a paper in Nature Medicine, found heightened immune dysfunction in tumors of obese melanoma patients. But Gibson, who just published a study relatively recently, found limited changes to systemic and local immune profiles specifically in clear cell. So I think it depends on, I think it's important not to lump all the tumors together. I think it's important to really see what's going on inside your each tumor specifically, tumor type specifically. The last piece of evidence that when we tried to figure out what was going on with tumor biology differences by BMI is that we leveraged something called the Clear Code 34 code set. This is This is a molecular signature involving 34 genes that was discovered by a group in UNC in 2014, and it's been extensively validated. And what it does is it takes 34 genes, and they're listed here, and splits them into CCA subtype and CCB. And the way I remember these is CCB, I think of as standing for bad. So CCB tumors are more aggressive. They are characterized by genes related to epithelial to mesenchymal transition, cell cycle, and wound healing. And then there's CCA tumors, which are less aggressive, and they're characterized by hypoxia, angiogenesis, and fatty acid metabolism. The research that's been done suggests that CCB robustly and independently is associated with increased cancer-specific mortality. The hazard ratio is 3.3, and for overall mortality, 2.5. So CCB is associated with worse outcomes and more aggressive genes that are expressed. Hackey et al. In 2016, working with Kim Rathmel, who was one of the people who discovered the Clear Code 34 assay. He looked at associations between BMI and Clear Code subtype. So he had 262 clear cell patients, and he found that obese patients were less likely to present with CCB than CCA tumors. Remember, CCB are the aggressive ones. So obese people tended to have more CCA instead of CCB tumors, and the odds ratio was 0.56. We confirmed this pattern in MSK data. These are unpublished data. These are 2007 clear cell patients. We found an odds ratio of 0.67. And this is something I wanted to mention is that Mark Perdue from NCI is doing a very nice study looking at etiologic heterogeneity and how different risk factors might be associated with risk of developing CCA versus CCB subtype. And what he's finding so far, our paper has been submitted and is under review, but we're finding that obesity is more strongly associated with risk of developing CCA subtype, and that's the less aggressive one. So taken together, our group is concluding that the BMI paradox appears to have a biological basis, that disease heterogeneity is influencing this paradox. Non-metastatic patients with high BMI may be surviving longer because they harbor less aggressive tumors. And for metastatic patients with high BMI, they might be doing better because they're given a treatment that targets the pathways that are upregulated in the tumors, like the angiogenesis pathway. Another sort of take-home message about this phenomenon so far is that normal weight patients might not be so normal or so healthy. So it's kind of the reverse of what I just said, which is just, but I'd like to draw people's attention to it's fine to be focusing on like what is good about being obese, but we have to remember that everybody has cancer, so it's not really a good thing. It's that there's some characteristic of the normal weight patients which is making them not do so well in these analyses of survival or on treatment. So compared with patients with high BMI, normal weight patients seem to have a feature, which are these more aggressive CCB tumors that is more lethal. And so this might be a form of collider bias, but right now we don't know what the risk factor is that causes that aggressive tumor. So again, this is why I think Mark Perdue's paper on etiologic heterogeneity is very important. So I'd like to shift gears now and also emphasize that high BMI is itself kind of confusing. So what does that mean? And I think that we need to be thinking about body composition. And probably to this group, you already know that CTs can be used to derive new and more specific information about different tissues that a person has. So BMI is a crude marker of body size. And again, what's nice about kidney cancer is that they have CTs and MRIs that are taken as part of routine care. Biopsies, I don't know if I mentioned this in the beginning, but biopsies are relatively uncommon in kidney cancer. And so all the surgeon has to go on are these images where they're trying to figure out where the tumor is, but now we in research can leverage these to read for body composition. So there's plenty of them. They're existing, they're pre-treatment, and they can be used to derive more specific body size features. These include skeletal muscle cross-sectional area and density, visceral adipose tissue, subcutaneous adipose tissue. And so for those of you who haven't seen CT scans before, This was taken from a paper by Antun in 2014, where he shows two very interesting patients. Patient A has, this is the cross-sectional image. This is the vertebrae at L3. The yellow is visceral fat, the red is muscle, and the blue is subcutaneous fat. And so patient A has a lot of subcutaneous fat, a lot of visceral fat, and relatively smaller amounts of skeletal muscle tissue compared to patient B, who has a lot more skeletal muscle, less and less fat. These people have the same exact BMI. So I think it's important to really distinguish what kind of tumor, what kinds of tissues these patients have, because we all know that adipose tissue and muscle are biologically active organs. So what's known about body composition in relation to clear cell? We know that a couple of things. The main analysis that's been done to date has been a really nice systematic review and meta-analysis by Helena Furberg from the Netherlands. It was published in European Urology Focus. What was great is that she was able to do a meta-analysis among metastatic patients treated with targeted therapy. And that's important because clinicians want to know if this could be a potential risk stratification variable or a treatment selection variable. She was able to meta-analyze 6 studies with 559 patients, and she found that low versus high skeletal muscle was associated with increased risk of increased mortality risk. She also meta-analyzed 4 studies with 429 patients total, also showing that low versus high skeletal muscle density is associated with a higher mortality risk. Unfortunately, so basically the take-home message here is that muscle seems to be playing an important role. There was inconsistent associations with visceral adiposity and also subcutaneous adiposity. And most notably, meta-analysis could not be performed for non-metastatic cases because there was such a wide discrepancy. There weren't very many studies in the first place, and there was a wide discrepancy in terms of cut points and exactly what they measured and when they measured them, so that was not possible. She did not include any studies of body composition and outcomes in patients treated with immunotherapy because this was at a time when immunotherapy was not an approved treatment. So now focusing on localized kidney cancer, so non-metastatic kidney cancer. One of the largest studies that was included in Helena's paper was done by Sarah Suka in 2016, published in the Journal of Urology. This is the largest retrospective cohort of body composition and mortality. So she looked at 387 non-metastatic kidney cancer patients treated by nephrectomy. She defined low muscle as that consistent with the international consensus of cachexia definition. That was published in Lancet Oncology in 2011. I think this group probably knows that there's a lot of debate, and it's not clear how exactly we should be defining these body composition features. But this was something that she did just as a starting point. And as what she found was that 47 patients diagnosed with localized disease had low muscle. And so if you think about that, that's a lot. That's almost 50% of the patients have low skeletal muscle. And when she looked at how low skeletal muscle was associated with mortality, she saw that similar findings as what Furberg saw in metastatic patients, that low muscle was adversely associated with risk of dying. So overall mortality hazard ratio was 1.4 and cancer specific was 1.7. She adjusted for age, sex, symptoms, BMI, performance status, and also these different tumor classifications. After reading the literature and understanding that skeletal muscle might be important, we decided to take the same approach that we did in the JNCI paper to try to see how body composition features, specifically skeletal muscle, since that seems to be emerging as the main one for kidney cancer. We thought we'd look at how if there were tumor differences by skeletal muscle. So for this, we turned to another NCI initiative. This one is called the Cancer Imaging Archive. It's a free and publicly available resource. I encourage you to check it out. It is organized by cancer type, and what it does is it's an initiative for radiogenomics. So different groups and institutions are submitting CTs, MRIs, ultrasounds, because there's a whole field of radiogenomics that are trying to figure out if we could, allowing radiologists to be able to predict what kind of biologically active tumor has based on what the tumors look like in the scans. But these scans can also be used for, theoretically, for for body composition analysis. When we did this, we learned a very important lesson. So we thought this would just be very quick. We'd just go to the website, download the information, and run our analysis. And we thought we could do that on the entire TCGA cohort. But what we realized, and the reason we realized this is because the radiologist at MSK was in charge of collating all of the variables for the TCIA and all the images from participating sites to the TCIA website. And what he told us were that the dates that are listed on the CTs are scrambled for anonymization purposes, which is important to protect patient privacy. But it becomes a really big headache when you're trying to make sure that you're grabbing the right scans to interpret for body composition and make sure that you're trying to get the CT that's taken around the same time for everyone. They told us also that images are submitted that come from inconsistent times during their treatment trajectory. So for some patients, you'll see their pre-treatment scan. For some patients, you'll see their post-treatment scan 6 months later, 1 month later, 12 months later. Sometimes it's 6 months before nephrectomy happens. So it's highly variable, and it's very difficult to select and be really careful about which CTs you're getting. Height as a variable is also not listed there. So if you're going to divide by height squared for any of your body composition variables, then that's not possible using this particular resource just as a user. So I think that in order to leverage this resource, and it's a great resource, you need to get in touch with the contributing institutions to properly select the CTs, because you're going to need their date key to backtrack and figure out when these CTs were available. And for this, you're probably going to need IRB approval. We were able to do this, but instead of having a sample size of over 300 patients, we could only get a sample size of 76 clear cell patients. And these were the ones that were treated at MSK, so we knew exactly when their image was taken, and we interpreted it for skeletal muscle using Flysomatic software. We used the same categorization for low muscle that Suka used, because we were trying to see if we could help explain her association. We classified them into CCA and CCB. Remember, CCB is more aggressive. So here's our 2x2 table. CCB subtype is across the top. 42% of cases were CCB. 40% of cases in the TCGA cohort of MSK patients were low skeletal muscle. And the adjusted odds ratio, after adjusting for age, sex, stage, and BMI, was 9. And it was significant. Even though it's a small subset, you can see that there's a very striking association between skeletal muscle and subtype. So in these more aggressive tumors, you are much more likely to see a low BMI, a low SMI, and in the CCA subtype, you're much more likely to see patients with higher muscle. So now I just want to spend the rest of the time talking about, that was sort of the background of what we're seeing so far, and that was the pilot data that led to the design and submission of our R01 proposal. So we're calling it the RESOLVE study because we're trying to resolve this BMI paradox. It's a retrospective cohort study, as I mentioned. It has 1,200 clear cell patients from MSK that we're analyzing. The eligibility criteria we decided to be pretty strict. So these are patients who are having their first nephrectomy, either partial or radical, between 2000 and 2019. They are non-metastatic, so we restricted it to just patients who get nephrectomy, so stages 1 through 3. There's no prior cancer diagnosis because we worried that if people had a prior cancer diagnosis and treatment, that could already be affecting their body size variable. Patients are adults, so ages 18 to 85. The average age of diagnosis is about 65 years old. They consented to future research on specimen protocols. We have existing pre-surgical CT scans that include L3, and then we have archived representative tumor blocks because MSK does a really nice job of, after a patient consents to future research, we bank a portion of their tumor that's not used for clinical management. And we also set a criteria that we had to have a minimum follow-up time of 6 months for each patient. Here are the aims of our study. There's 3 of them, and we just, the first one is really trying to understand the interrelationship between BMI, body composition, and molecular tumor subtype. So we're going to see if we can validate some of the initial signals that we saw leading up to the pilot data leading up to the grant. Aim 2 is looking for body composition variables that are independently associated with prognosis after accounting for tumor subtype. So to do this, we're doing mediation analysis, and we're also stratifying our large sample size into CCA and CCB tumors, and then looking at how body size is associated with outcomes in each one of those categories. Aim 3 is a more sort of biologic aim, and this is looking at transcriptomic signatures of body composition and of prognosis. So we're hoping that these aims will clarify associations between pre-treatment body size and clinical outcomes, and also provide new insights into the underlying biology of adverse body composition features. Our workflow is that we are linking clinical data with tumor profiling with CT. So the clinical data are being downloaded and reviewed. We've got access to variables like demographics, BMI, smoking history, whether they presented with symptoms, comorbidities, stage grade, tumor size, Karnofsky performance status, vital status, and cause of death. We also have CKD measurements like EGFR, but we don't have a lot of information on pre-diagnosis weight loss and physical activity. We are retrieving archived tumor blocks from the MSK Biobank, and what we're doing here is our pathologist is reviewing and sectioning representative FFPE blocks. We acknowledge that there may be intratumoral heterogeneity. PD tumors can be very large, so we're selecting one block. If the patient has a tumor that is, we're doing one curl, and we're making one 10-micron curl. If they have a tumor that is less than 7 centimeters, and we're taking two curls from different regions of the tumor if their tumor is greater than 7 centimeters, because it's been shown that if a tumor is greater than 7 centimeters, there could be more heterogeneity. And we're profiling that, so if we get, for example, discrepant findings, and in a larger tumor, we might find that one tumor is CCA and one is CCB, we'll call it a CCB, because that's the more aggressive lesion. We're profiling tumor specimens at UNC Chapel Hill on a custom nanostrain code set. These include genes from the clear code assay, as well as the immune pathway, inflammation, angiogenesis, hypoxia, and metabolism. The people involved in selecting these genes, one thing I didn't mention here, which I forgot, was David Thomas, who is studying the biology of sarcopenia and wasting, also contributed some pathways involved in those, and so our custom panel includes those as well. So we're QCing the data, classifying them into the subtypes, and also the relevant pathways. As for the CTs, we are retrieving them with a partnership from MSK Radiology. They are anonymizing them first, and then we are manually reviewing them for eligibility. And so Marina and Mike have really been instrumental in making sure that we follow strict protocol for eligibility. And we're only using scans that were pre-treatment, pre-nephrectomy scans that were taken up to 2 months before nephrectomy. They have to have a scan available at L3. And scans are ineligible if there are bright artifacts, rods, or tissues that are grossly cut off. So I just thought for fun I would show you an ineligible CT. This you can see, so here we have the vertebrae to orient you, and this person has, it looks like a lot of subcutaneous adipose tissue. Here's their muscle, but there's this bright artifact here on the left, and this means that the person was sort of pushed up against the slide, against the scanner, and so it created this bright artifact. And so for us, this is problematic because now they've got, you can see that their subcutaneous fat is over here, but it's completely cut off over here. And in fact, it even goes into the muscle. So this is something that you can put this scan into an automated program, for example, and you will get information out, but it will be inaccurate because these people, you need to do your manual review before you read these scans to make sure that you're taking care of all these issues that could be lurking in your CT. It's also important to note that if contrast was used, Mike Paris, one of our team members, published this paper in 2018 showing that contrast influences radiodensity, especially, this was a study in clear cell renal cell carcinoma. So we are taking great care to note we're trying to grab non-contrast images, but we can, even if we do have contrast, the patient only has contrast images, we feel okay about using their cross-sectional areas. And here I would just like to introduce something called Automatica software. So again, Mike Paris and Marina's lab from the University of Waterloo has developed open source software that can be used to automatically read CTs and segment them into these body composition compartments. Please feel free to contact Mike. He said it was okay to put his name and his contact information on this slide. And please also refer to this published paper that came out last year in Clinical Nutrition that describes the program, that we're using it, and it's working great. I should also say that I loved Faisal's presentation last month, and he and I are talking about how it would be really interesting to see if we could use his program for these kidney cancer patients, depending on what scans we have available, because I loved hearing that his program goes above body composition to look at other features that might be important. Okay, so the progress to date. We have identified all of our patients. We've finished reading the scans thanks to Mike's program. We've retrieved over 500 tumor specimens. We've been a little bit delayed because of COVID, but thankfully this is a retrospective study, so we haven't been set back that far. UNC has extracted about RNA from about 200 curls. We are in the process of ordering our custom nanostring code set, and we have reviewed and QC'd the clinical data. We are also in the process right now of requesting the cause of death data from the CDC. This will be important, especially last year and this year, because now patients could be dying of COVID, and we want to make sure that we label that in our data set. I think that the strengths of our results study is that it'll be the first study to link body composition with gene expression to inform clinical outcomes, or to look at how they're associated with clinical outcomes. It has a very large sample size to allow for for subgroup analyses. It's focused on clear cell, which is the most common and lethal kind of kidney cancer. But a very big limitation is that we're only evaluating so far pre-treatment CT scans at baseline. And we also don't have data on physical activity, hand grip strength, or diet, other things that are important to have in order to really understand and phenotype these people. Also, the generalizability of our population is limited to white. That is the bulk of our cohort, 90% of them. I think it's really important that we do these types of analyses in other racial and ethnic groups. And also look at non-metastatic, it's a limitation of the RESOLVE study is that we're focused on non-metastatic patients. So to address that last part, we put together and were awarded an administrative supplement for this grant last summer, and this allowed us to expand the RESOLVE study to include metastatic clear cell patients treated with immunotherapy. So I went to the medical oncology department, and talked to Martin Voss and colleagues, and we were able to put together an observational cohort of 250 patients treated exclusively with immunotherapy. They have body BMI and CT imaging at CT start, so we can read their body composition using Automatica, which we've done. And here the outcomes are overall mortality, treatment response, and so in summary, I hope I've presented some data that supports that the BMI paradox has a biological basis. And this is something that you might, those of you that are studying different types of tumors, I think this is a really important thing to consider. So if you're seeing the BMI paradox in melanoma, or which they are, and in lung cancer, which they are, and lots of other cancers, I think it's very important to think about tumor biology. And if your tumor of choice has different subtypes, consider stratifying by those and seeing how the associations differ. We're seeing that patients with high BMI have the survival advantage when they're compared to normal weight patients in these case-only analyses. And we think that this is driven by the differences. The apparent advantage of high BMI patients is really driven by tumor biology. So it's not appropriate to advise patients to fatten up to improve clinical outcomes. We have a lot of work to do to figure out how adipose tissue specifically and adipose tissue in combination with skeletal muscle influences survival. It's also not clear yet whether BMI should be used for treatment selection. I've been seeing a lot of papers recently saying that BMI would be a great stratification variable since high BMI patients do better, for example, on targeted therapies. But in my next slide, I'll just say a few words more about that. I think that existing CT scans can also provide new insights. We can refine body size into more specific tissues. That is incredibly important since they have different biological properties and can do different things. There's really exciting data coming out showing that muscle in particular can influence the immune system in the systemic environment and also in the local environment around the tumor. To date, low muscle is emerging as the independent prognostic factor, but I'm sure it's going to be really important to think about how adipose tissue also factors in. And we're hoping that the RESOLVE study reveals novel prognostic factors at treatment start, and then also, pre-nephrectomy, and then also how tumor transcriptomic signatures are associated with these body comp features. So for my last slide, I just want to talk a little bit about the future directions and where I think, what is needed in the work that my colleagues and I are doing. And these might apply to what you're doing as well. I think it's critical that, I think the RESOLVE study is an excellent and very important first start where we're looking at baseline CT and looking at how that is associated, because clinicians do want a non-invasive way to predict prognosis, and body composition might be that, especially if it's a proxy for tumor biology. But I think it's really important that we describe body composition changes after diagnosis and figure out how they affect prognosis. So, for example, we are in the process of getting serial CT scans on our patients to determine how much wasting happens after nephrectomy, and also what happens after treatment in the metastatic patient. Another thing that I think is really important is we have to figure out and comprehensively characterize these people with adverse body composition features, and we need to go beyond the tumor. So how is weight history affecting how they present at baseline? What about their physical activity levels, and what about their cardiovascular health? For example, one way to think about it is, I think an important question still to be resolved is that we're seeing 40 and 47% of patients with low muscle at the time of diagnosis. Is this because they're inactive, or is it because they have aggressive tumors? So there's not a perfect correlation with aggressive tumors. So I have a feeling it's both. And I think to inform future interventions, we'll really have to unpack that. And finally, to determine if body composition is predictive as well as prognostic, I'd like to refer you to an excellent article written by Carla Ballman from Cornell. She's a statistician. She published it in JCO in 2015. And she explains the difference between looking at a prognostic factor versus predictive one. And what she is emphasizing is that you really need to test for interaction. You really need a clinical trial that has 2 treatment arms and body composition on everyone to figure out which body composition group benefits from which treatment. So that's something to think about. And our hope is that we can use body composition as a novel stratification variable, and also possibly modify it to improve treatment outcomes for the growing number of kidney cancer patients. And so I think with that, I'll stop and take any questions. Thanks.
Great. Thank you so much. Great talk. Lots of comments saying great talk. Here's a question about the RESOLVE study. Do you have a sense of how your RESOLVE cohort compares to the general population? One could suspect that patients treated at Sloan Kettering may either have a tumor that's biologically different, maybe more aggressive, or may be different socioeconomically.
Yes, that is always a concern when we're doing our analyses at MSK in terms of that's the generalizability issue that I was mentioning, and definitely it is. People who tend to be treated at MSK are often more educated and are higher SES and are more often white than other racial groups. So I think that the good side of that is that, as I mentioned in the beginning, clear cell, which is the most lethal and aggressive subtype, more often occurs in men and in people that are white compared to other racial and ethnic groups. So in some sense, it's good that we've got this homogeneous cohort and we've got 1,200 people with those features. And the other thing that we're doing is, it's true, I think the part about are we seeing more aggressive cancers, yes. I mean, it's true that at MSK we tend to see very problematic and sort of scarier tumors in a lot of other disease types, but in kidney cancer, we're still seeing these not indolent tumors, but these incidentally detected ones. So I think the problem is only generalizable to whites. I don't know that the socioeconomic status is going to influence things, because I think the distribution of the type of disease we're seeing is very similar to the population at large.
A couple of questions about racial differences. Do you know why there's a higher rate of kidney cancer among whites, and what about Hispanics, and both white and Black Hispanics?
So for this, I mean, it's a really interesting question, and I mean, as all of my research has shown, I have been focused predominantly on studying clear cell that occurs more often in white. So histology is a big thing here. So even though kidney cancer sounds like it's one disease, it's actually several different ones. So clear cell is the most common histology, and then there's also papillary and chromophobe, and those are less aggressive. And African American people tend to have papillary tumors, and which tend to be less aggressive, and women also tend to have these other subtypes, basically these non-clear cell ones. And I think it's incredibly important to study those. I know that there's a great initiative at Vanderbilt going on. Lauren Lipworth is doing a lot of racial disparity research in kidney cancer in the Southern Community Cohort Study. So I don't know what is happening. I also know that the incidence of kidney cancer is increasing in Latinos. And one thing is that that people have talked about is that we are seeing this increasing incidence of these small tumors, and we're also seeing an obesity epidemic happening at the same exact time. So it may be that there are body size differences in the risk factor profiles that make them more likely to develop these tumors. But it's a little unusual. You would think with that logic that they should be more likely to have clear cell tumors, but in fact we're seeing their different histologies altogether.
Great. I know you're focused in adult patients, but does this BMI paradox also exist among pediatric populations?
I have no idea. That is a great question. I do not know. I know there's kidney cancer in children called Wilms tumor, but that's very rare. And honestly, I don't know anything about BMI in those patients.
Great. We have a question saying that they've seen the same relationship in lung cancers, and some are arguing that it's protective against cachexia. Do you have thoughts on this argument?
Well, I think that's what is really important with the RESOLVE study. I think I can't answer that yet, and that's kind of what I was saying, is that some interpret the association to be causal, that people with high BMI can withstand the metabolic demands of cancer better, and that there might be some merit to that, especially since there's evidence that there's a crosstalk going on between fat cells and tumor cells, and fat cells might provide the fuel for the cancer. So on the one hand, obesity, might make provide the tumor cell with more fuel, which would make it divide more rapidly. On the other hand, it might provide a food source that's local, so it doesn't have to go anywhere, making the tumor less aggressive and less likely to metastasize. So I think there's a lot of biology that needs to be done to unpack that first, and I think the local tumor microenvironment is a great place to study that. But I also think that it's really important, that's what I was saying, is that we need to look at the trajectory of body composition over time to see, who is wasting and why are they wasting, and are they wasting because of the tumor biology that we see at baseline.
Great. In your tumor expression data, are you just focused on the primary site? Have you also looked at the metastases?
That's an awesome question. And the answer is no, we've only looked at the primary site. So that would be a great and important thing to do, is look at the metastatic lesion to see what those tumor features are. So in the slides that I presented that showed gene expression patterns in metastatic patients, the patients were metastatic, but their tumor that was used for the profiling was their tumor, their primary tumor that was taken out as part of their care. But I think it would be important to focus on and try to figure out how is the tumor changing, just like how is the body composition changing, how is the tumor changing as it progresses.
Okay, we are running out of time, and anyone who I don't get to your questions, I will have Helena respond to you. But let's see, how about, are there thoughts on targeting FASN? Are there ongoing studies that you're aware of?
I am not aware of any studies. And this is where, I think some of my colleagues might know more about that, especially the clinical ones, the clinicians and the physicians. Yeah, so I'm not sure.
One last question. What about short-term outcomes, surgical outcomes and complications? Have you added that to Resolve?
We are in the process of doing so. So our primary, that's also an important point. Our primary outcomes are survival, cancer-specific and overall, and then and progression-free survival. But it's very important, and this is kind of an advantage of being doing a hospital-based study and at a single institution, is that we have access to everything in their medical records, so we can look at complication rates and things like that. But in Resolve, again, kind of what's interesting about studying kidney cancer is that there's not going to be a lot of treatment effects or toxicities or anything like that, because they're not getting any kind of therapy. So yes, we definitely plan on analyzing those as well.
Great. Well, thank you so much for joining us today, and thank you to all the attendees who are on this call. Next month, February 11th, we'll have Dr. Michael Sawyer from the University of Alberta talking to us about treatment-related toxicities. I hope to see you all then, and thank you for joining us today.
Thank you.